Today we'll talk about an important topic in applied in genetic engineering and its forensic dedication of criminals. First of all, thank you for Dr. Nisreen and Dr. Dalia, and we give them our gratitude for all they have done. At the introduction, we will talk about the definition of forensic chemistry. Forensic chemistry is a branch of chemistry that deals with the application of analytical techniques to solve criminals' cases. Also, it analyzes evidence from crime scenes. History of forensic chemistry back to 18th century France when chemists were asked by authorities to help them solve poisoning and other crimes using their knowledge. Where conventional methods failed to provide any clue about the identity or a suspect or victim or object involved in certain crime scenes. So they need to protect an idea to help them with that. And forensic chemistry were the best idea to deal with. Now we'll talk about technique of gene. It have a three important points. One of them is PCR. PCR is a common molecular biology technique used in most applications of gene technology. For example, DNA profiling. The second point is DNA profiling, genetic fingerprinting. It enables scientists to identify suspects for a crime, except for identical twins because they have repeating short non-coding region of DNA. These repeats occur at the same position on chromosomes, but the number of repeats varies between individuals. DNA can be found in skin cells or blood or hair or salvia. To create a DNA profile from the DNA being tested, we need to follow this sequence. Step number 1. Obtain the DNA which can be extracted from the roof of a hair or a spot of blood. Step number 2. Increase the quantity of DNA by using PCR. Step number 3. Use restriction. Step number 4. Separate the fragments using gel electrophoresis. The final step, add radioactive. Criminal investigations. DNA has been used by forensic scientists to identify suspects of crime. In this example, we have one victim and three suspects. Samples of body cells or fluids like blood or hair are taken from the crime scene or victim body, and we compared it with the samples from the three suspects. DNA will help us to identify which one is the real criminal. Advantages of forensic chemistry. The first one, forensic chemistry can help us to detect if the fire is set intentionally or not. The second one, it helps us with the suspicion list and make investigation easier. Also, it can make identification of particular poisoning. Advantages of forensic chemistry. First one, long and compressor procedure involving consistent working for hours in DNA analysis are found to be unethical processes. So a person might face a serious problems. Also, it can deteriorate the health conditions of an individual. Finally, the reagents and equipments used are expensive. Now we'll talk about how is genetic engineering used in forensic. DNA profiling is mostly used by forensic scientists and crime lab techniques to identify criminals and victims using track evidence like hair or skin samples. To produce a DNA profile, scientists compare sequences in the genome that vary from person to person. Advantages of genetic engineering in forensic chemistry. First one, small amount of evidence can be amplified and used to produce accurate profiles. Also, majority of crimes committed in today's society. The disadvantages of genetic engineering in forensic chemistry. The cost of establishing and maintaining are increasing. Now we'll talk about the future aspects for more achievements and developments. The first one is perceiving boon. Phone holds a reservoir of information that can be key in forensic investigation. The second one is a simpler way to view fingerprints, so the criminal investigations will be more easier. The third point, the true power of Proteus for forensic, which can sometimes answer questions that DNA can't. And the final point is handheld device, and it also called mass space pen and it easy to use.